It starts from the glucose and end with the pyruvate. So these 10 steps, if you observe, there are some steps having some coupling of reactions. For example, glucose is converting into glucose 6-phosphate. At the same time, ATP is converting into ADP. So try to understand these coupling reactions. And here, fructose 6-phosphate to 1,6-fructose bisphosphate is converting. At the same time, ATP is converting into ATP. And here, glyceraldehyde 3-phosphate to 3-bisphosphoglycerate is converting. At the same time, NAD become NADH. And 1,3-bisphosphoglycerate to 3-phosphoglycerate, at the same time ADP into ATP. And phosphoenol pyruvate to pyruvate. And at the same time, another reaction is going on, ADP plus inorganic phosphate, it's giving rise ATP. So these are all called as coupling reactions. Okay, so we have remaining all we have learned, and the each step, what is going on, what is the kind of reaction is happening, and which is the enzyme catalyzing this reaction, and energetically what are the steps are involved. Everything we have understood. Okay, so with this, let us go to the second topic in this course, in this glycolysis, that is little bit more we can understand the summary of it, and regulation, and as well as significance. Okay, maybe it is not mentioned anywhere in the slide what you are going to learn, but at the end you can see this slide what you have learned. Okay, so at the beginning, first let us have a idea what is the significance, and energetically what are the things happening. See, this is the definition: set of reactions that oxidize glucose to pyruvate under aerobic or to lactate under anaerobic. This is very clear, right? And aerobic conditions, it will go until pyruvate, it will stop. And in case of anaerobic, it will go until the lactate. Okay, so can someone of you give your answer? Why in anaerobic conditions it need to form lactate? Any of you? Okay, so then if you don't have, maybe I will ask in randomly. So Dharmega Loshini,
Yes, sir.
my question is, why anaerobic condition it's not ending with the pyruvate, it is extending unto lactate? Or anyone else? Belnantho, Daryl Nathan, Arvind Ganesh.
Yes, sir.
Yeah, the question is why anaerobic conditions it is forming lactate not until the pyruvate. Felicia,
So is it
yes,
because they want to recycle back the NAD?
very good, excellent. See, in anaerobic conditions, if you stop with the pyruvate, what happens? Pyruvate, the NADH which is produced in uh, glycolysis, it is not going to be processed further. It is going to be accumulated NADH, and only ATP which is produced. Some of the ATPs it will be can be utilized, but the whole glycolysis is going to be arrested. So no more glycolysis. So there will be no energy will be further continued. because after the glycolysis, I think you may be having a rough idea that it will enter into the Krebs cycle, and where it is also required NADH need to be will be produced, and that need to be processed for the electron transport chain for this, the respiration, that is oxygen required. So if you if the pyruvate is not converted into lactate, what happens? Accumulation of pyruvate, accumulation of NADH, but it's not utilizing. And in ATP will be not much will be there, just few, whatever the glycolysis occurred, whatever the NAD was available, all NAD will be converted into NADH. Until that, it will produce ATP. After that, so two things: one, it is going to be toxic because accumulation of NADH, then arresting of the glycolysis. So it is not good. So it need to be go for the converting the pyruvate into lactate. So thus, what happens? The NADH will be converted into NAD, which is needed for the another cycle, another pathway of glycolysis will be available, synthesized, and also NADH accumulation also it can prevent. So thus, anaerobic condition, it must undergo the conversion of pyruvate into lactate. So, for example, when you are uh, running very fast and continuously, or walking or running especially, so what happens, the muscles will take all the muscles, muscle cells all undergo this glycolysis, and it will produce the ATP and produce the NADH. Then NADH need to go for the aerobic respiration, that is in the electron transport chain, but it is not having that much time, but because that not even that much oxygen, because when you are taking the oxygen, that may be not sufficient to convert everything into ATP. So those muscles slightly will be having anaerobic condition during that time. So what happens that the leg muscle cells will go for the anaerobic glycolysis, and then whatever the pyruvate it was there, it was produced, it will go and undergo anaerobic. So advantage of it, instant cache, instant ATP will be produced, which is needed for immediately. Okay, so that is one advantage. And sufficient oxygen was not there during that no severe exercise. So because of that, anaerobic reaction it choose. So thus accumulation of the lactate will be there in the muscles. So that's why the next day, after few hours of the next day, so you will have the cramps, the muscle cramps, because of the excess accumulation of the lactate in the muscles. So what need to be happen? That lactate again need to be processed further for some other process that we are going to learn in the maybe in the next class. So what happens to the lactate? That is entirely different reactions. Okay, so that is what the importance of the anaerobic glycolysis. Then where it occurs in cytosol of liver and extrahepatic. Hepatic mean liver, extrahepatic mean other than liver. So it will occur liver as well as other than liver. So why it is stressed on liver? So liver is the major, no, the tissue uh, to performing the glycolysis, but everywhere in the cell, any in the body, it will occur. So what is the summary of this reaction? Glucose plus NAD, 2 NAD plus 2 ATP plus 2 inorganic phosphate gives 2 NADH plus 2 pyruvate, 2 lactate ATP, then 2 water plus 4 protons. This is the summary of this. In this, some of the things what we have understood, there is a couple reaction. For example, phosphoenol pyruvate, which is be converting into pyruvate. So it will be added water molecule and inorganic phosphate will be released during this reaction. And for that, free energy change is 62 kilojoules per mole. So energy will be released during this reaction. So phosphoenol pyruvate is having more free energy. Pyruvate is having less free energy. So whatever the excess energy it is having the PEP will be released during the time. How much is that? The free energy change is the one free energy of the pyruvate minus free energy of the PEP, phosphoenol pyruvate, is called as free energy change, Gibbs free energy change. And that is 62 kilojoules per mole. And during this time, one more reaction is happening. ADP will be converting into ATP by adding inorganic phosphate, which required 30 kilojoules per mole. So that means ADP having less free energy, ATP having more free energy. So will it occur randomly or spontaneously without any boosting? It is not going to be happen, right? Because some energy required it. ATP needs some energy from outside to add to it to form ATP. ATP having more energy. So these 30 kilojoules are needed. From where it is going to come if it is need to be spontaneous? So it is not going to be spontaneous. It needs some outside energy. How it is coming? It is coming from this conversion. PEP to pyruvate, which is 62, it is releasing. Out of this 62, 30 kilojoules will be taken up to form this reaction. So this is going to be happen as a coupled reaction. It's called as couple. So when it's going to be couple, the two reactions are the same time happening. So energy released in one reaction is utilized by another reaction. At the same time, these both reactions are catalyzing by one enzyme, that is pyruvate kinase. That is also important. So what is the summary? PEP plus ADP is giving rise pyruvate plus ATP, pyruvate kinase. The, the net change is 32 kilojoules per mole will be released. Still extra energy will be released. So that is the coupled reaction which is happening to produce the ATP. So which is not a coupled reaction. For example, so PEP is giving the pyruvate and where free energy is releasing as 62 kilojoules. And glycolysis first reaction, glucose plus the inorganic phosphate, that is glucose 6 phosphate is forming. And where 14 kilojoules are needed. And actually what we learned during this reaction, ATP utilizing and ATP is converting to ATP, and that is the energy it is utilizing. But instead that will it be happen like this, PEP to pyruvate where 62 is releasing. So can the reaction take this energy to form glucose to glucose 6 phosphate instead of ADP to ATP? Definitely no. This is not a coupled reaction. The reason is different enzyme catalyzing these reactions. That is the main reason. That's why ATP is converting into ADP where 30 kilojoules are releasing. So the reverse reaction, that is ATP is hydrolyzing, and during that time glucose will be forming glucose 6 phosphate. So for that 14 kilojoules are needed. So out of 30, the 14 will be utilized, and still glucose to glucose 6 phosphate 16 kilojoules are released. Okay, then pyruvate conversion. So what happens to the pyruvate which is formed in the end of the glycolysis process? It will undergo three kinds of reactions. In case of oxygen present, so pyruvate will be converted into acetyl coenzyme A because NAD will be utilized during this time to form NADH. So thus oxygen is required. So aerobic conditions, it will undergo to form its acetyl coenzyme A, and then it will further process to enter into citric acid cycle. Or pyruvate will convert into lactate. So that means the anaerobic condition and NADH is utilized and NAD is formed, and then lactate is forming during this time. In some of the microbes, what happens? So pyruvate will also convert into acetaldehyde after removal of the carbon dioxide, and then NADH will be utilized to form ethanol, where you can see the fermentation. So ethanol will be formed. Okay. So metabolic significance. Give me just 1 minute. So metabolic significance of the glycolysis. This is the principal route of glucose utilizing the to produce acetyl-CoA and oxidation in citric acid cycle. So this route is very important to go enter into the citric acid cycle where it will be having producing a lot of other energy. So this is the main route. You cannot exclude this route in the carbohydrate or glucose metabolism. And it will provide 8 ATPs in aerobic and 2 ATPs in glycolysis. So a lot of energy is there, and provide ATP in the absence of oxygen. See, that is very important. So when even the no oxygen, so the aerobic condition still ATP, which is needed for the basic energy purpose, it is you can provide, it will provide. So thus allows skeletal muscle to get energy when aerobic respiration is insufficient. That is what just now I explained to you. When aerobic respiration during the severe or aggressive running, during the time, if it is not sufficient, the oxygen, so it will still get ATP to run by utilizing the whatever the glucose present in your cells. It will be utilized and gives the instant ATP to utilize. And the RBC, 90%, eye tissues are 84% of the energy is derived from glycolysis. This this is another very important red blood cells. The main getting energy for RBC is glycolysis, because it is not having other parts of some of the components of the cell, like nucleus and mitochondria kind of thing, where it will not undergo those kind of aerobic respiration. So glycolysis is the major pathway to get the energy for red blood cells, which is very important cells in our body, isn't it? So conversion of 1,3-bisphosphoglycerate to 2,3-bisphosphoglycerate, decrease in oxygen affinity, and shift the oxyhemoglobin dissociation curve to the right. This point I will try to explain in the further today's class. So where it will have a very important role in the respiration and to carry the oxygen by the hemoglobin. Glyceraldehyde 3-phosphate used for fatty acid synthesis. So glyceraldehyde 3-phosphate is one of the intermediate component which is produced during the glycolysis process, is the substrate for the TAG synthesis. So thus this is another important of the glycolysis, right? Modified reversible pathway is called as gluconeogenesis. So that we will be seeing in the next class. That means the glycolysis which is produce the pyruvate from the glucose, and the reverse of it, pyruvate to the glucose or lactate to the glucose, and this pathway we call as gluconeogenesis, that need to be undergo when the cells are not having sufficient glucose for the energy source. So that in detail we will try to understand in one of the class. Okay, come to this point where 1,3-bisphosphoglycerate is one of the intermediate in your glycolysis, and it's supposed to undergo with 3-phosphoglycerate, and ATP will be produced by phosphoglycerate kinase. This is one of the step, right? But alternatively, in especially red blood cells, what it will do, the 1,3-bisphosphoglycerate can convert into 2,3-bisphosphoglycerate. This is a special arrangement of the phosphate group, and which is called as 2,3-bisphosphoglycerate. And then it will be converted into 3-phosphoglycerate, and where the phosphate is releasing as an inorganic phosphate, not as ATP. So this is the mutase and phosphatase. In fact, both are having together one and big enzyme is present for this process. And then 3-phosphoglycerate is converted into 2-phosphoglycerate with the help of mutase. This is the normal reaction which is a part of the glycolysis. What is different? Normally 1,3-phosphoglycerate to 3-phosphoglycerate, 3-phosphoglycerate to 2-phosphoglycerate. This is the actual process of the glycolysis. And these two steps is different from the, please excuse me, I may need another 5 minutes, an important call. Thank you. Welcome back. So the extra difference is between the 1,3-bisphosphoglycerate to 3-phosphoglycerate, where it is happening normal glycolysis, but it is happening the same step in two different steps, that is converting into 2,3-bisphospho and 3-phospho, but ATP is producing normally, but in this reaction it won't produce. Where it happens, especially in the RBC. Regulation of glycolysis. So this glycolysis need to be get regulated. So not should not happen all the time and should not stop all the time, because whenever it is required, whenever excess glucose is there, need to be produce energy, it need to be conducted. I mean, it need to be, undergo. Otherwise, if it is having, a lot of energy and not required of any energy more, and it no need to have the glycolysis all the time, right? So it should have a regulation. So when to be happen, when to be stopped. So this is the called as regulation, and the key enzymes for this regulation are glucokinase or hexokinase. That is the first reaction to which is catalyzing this, or phosphofructokinase, and then pyruvate kinase. So these three enzymes which are catalyzing those three steps in glycolysis are the responsible for the regulation of this glycose. So control is allosteric control or covalent modification or induction or repression of enzyme synthesis. So how it is controlled and what enzymes are it is involving. So that is what this slide will show you. So if you still remember, what is the glucokinase function? Conversion of glucose to glucose 6-phosphate. That is the, or hexokinase also same thing. So that reaction is regulated. Then phosphofructokinase, what is the reaction catalyzing by this enzyme? 6-phosphoglycerate to 1,6-bisphosphoglycerate. That is another reaction catalyzing by phosphofructokinase. And pyruvate kinase, what is the reaction catalyzing by this reaction? This enzyme? Phosphoenol pyruvate to pyruvate. So among the 10 reactions of the glycolysis, these three reactions are in a single direction, only forward direction. That is the rate limiting steps in the glycolysis, whether to be undergo or to wait. So that information will be, conveyed by these enzymes through that the respective reactions. And how it will convey? There are three ways it will convey: allosteric control or covalent modification, or induction or repression of the synthesis. What is mean by allosteric control? Okay. Okay, these are the 10 reactions, right? Among these 10 reactions, what are the three reactions is controlling? So this the black thick arrow, where these reactions only in a single direction if you observe. Other all in a two directional. So either forward is possible or reverse is possible. And glucose to glucose 6-phosphate, or fructose 6-phosphate to fructose 1,6-bisphosphate, or phosphoenol pyruvate to pyruvate. These are the three reactions which are in a single or forward direction only, and these are the rate limiting, and these are the controlling the whole glycolysis process. And how it will control? For example, hexokinase or glucokinase, it will be inhibited by glucose 6-phosphate. What is the reaction catalyzing by this hexokinase? Glucose to glucose 6-phosphate, right? So that reaction, if it is having a lot of glucose 6-phosphate in the cells, do it require to undergo this reaction? Not required, right? So what happens? Glucose 6-phosphate bind to the hexokinase and stop that reaction. So that is called as allosteric control. Okay, allosteric control means the enzyme actually bind to the substrate and convert into product. That is the enzyme active site, it will bind. When enzyme bind to the another area with another molecule, which will change the structure of the enzyme, which will make the enzyme inactive, that is called as allosteric modification. So here glucose 6-phosphate, if there is excess, there is no need of converting glucose into glucose 6-phosphate, which is catalyzing by the hexokinase or glucokinase. So thus glucose 6-phosphate will bind to the hexokinase and stop that reaction. That is the inhibitor. So which is the activating molecule for this hexokinase? None. And phosphofructokinase, that is the another enzyme we have seen, right? This is the one. Phosphofructokinase catalyzing by fructose 6-phosphate to fructose 1,6-bisphosphate. What is happening here? Phosphofructokinase will be inhibited by ATP. It's sensible, right? If the cell is having excess of ATP, so no need to undergo that reaction, right? Glycolysis. So it will inhibit. If it is excess of citrate, citrate is where it will present after the glycolysis. Once the pyruvate convert into acetyl CoA, then enter into citric acid cycle. The first product is citrate. So that means when the more glycolysis is happening, more citrates will be forming. So when the more citrates are there, so it will give the information that phosphofructokinase to not convert the phospho fructose 6-phosphate to fructose 1,6-bisphosphate. Okay, and PEP, phosphoenol pyruvates. If excess of phosphoenol pyruvates also, it will inhibit the phosphofructokinase activity. In contrast, so ADPs or AMPs are very high. That means that means that ATP are less. Okay, then phosphofructokinase will be activated. Pyruvate kinase, which is conversion of pyruvate, phosphoenol pyruvate to pyruvate. ATP will be the inhibitor. There is nothing is activating. And yeah, phosphatase is another extra step which I have explained to you in the previous slides back. So that is AMP is the active inhibitor and ATP is the activator. So this is the allosteric activators of phosphofructokinase. So these are the plus mean, it is the activators, and minus mean, it is the repressors. So apart from that, it will go for the covalent modification. For example, phosphofructokinase, when it is dephosphorylated, so the phosphofructokinase is an enzyme which is a protein. So when it is don't have any phosphate moiety on the enzyme, it is called as dephosphorylated. That is going to be active form. So when you have a lot of energy inside the cell, so what happens? This phosphofructokinase will be phosphorylated, so that it will become inactivated. So once the enzyme have extra phosphate group on the enzyme, don't confuse this phosphate group from the glucose or fructose. It's not like that. This is phosphate on the protein. Which protein? An enzyme, phosphofructokinase. If it is having the phosphate moiety bind to it at particular position, it is called as phosphorylated, and it is the inactive form, so it is not going to be catalyze the reaction. So in case of dephosphorylated, it will be active. So thus the cell will give a signal to these enzymes whether it need to proceed the reaction or not by these covalent modifications. Okay, there are some senses which makes, for example, insulin. What it will do, insulin is a hormone, as all of you know. It will maintain the glucose metabolism, right? So how it will maintain? Insulin dephosphorylate the key enzymes like pyruvate kinase and phosphofructokinase. What is the function of insulin? Dephosphorylate. When it is dephosphorylated, what is the thing going to be happen? It is going to be activated forms. That activated forms mean it will undergo excess of glycolysis, and that the excess of glycolysis will occur. What happens? The glucose all will be utilized. That is how insulin reduces your glucose uh, molecules in the cell. You understand now what happens to glucose? It is a signaling pathway. So insulin not there are it is it's maybe not directly dephosphorylated, but there is a sequence of dephosphorylations occur once insulin bind to the cell surface uh, receptor. So then it's a sequence of modifications occur. Finally, some of the modifications like phosphofructokinase and pyruvate kinase will be get dephosphorylated. Thus, it will be get activated. Thus, glycolysis will be activated. Then glucose will be utilized. The glucose concentration in your cells will be reduced. So that is the function of the insulin. So what happens in case of glucagon, which is the contrast hormone to the insulin, either glucagon or epinephrine, it will act. It makes the phosphorylate these two these enzymes. Thus, it will be go for inactivated. Thus it will be it will inhibit the glycolysis process. That glucose concentration will be increased. So that is one of the way glucagons will increase the glucose molecules inside the cell. So that is about the glycolysis regulation. And then this pyruvate, so what happens to this pyruvate in aerobic conditions? What we have understand, in aerobic conditions it will undergo to form acetyl coenzyme A and forms the carbon dioxide. And for this reaction, the pyruvate dehydrogenase is the enzyme catalyzing this reaction. And for this enzyme, there are so many cofactors are there. I think you have understood, you have studied the enzymes and coenzymes, right? For this pyruvate dehydrogenase enzyme is a very big complex enzyme. It is having not one substrate, it is having multiple substrate, especially uh, it can be illustrated as three big enzymes together forms one enzyme, that is pyruvate dehydrogenase. And this enzyme will be having uh, coenzymes. There are 5 coenzymes. What are those? Coenzyme A, lipoate or lipoic acid, NAD, TPP, and FAD. So these are all 5 cofactors or coenzymes are required for, I think cofactors are needed for the pyruvate dehydrogenase enzyme to convert pyruvate into acetyl coenzyme A, where carbon dioxide will be released. So pyruvate is a 3 carbon containing molecule, and when it is losing 1 carbon in the form of carbon dioxide, so the acetyl coenzyme A is a 2 carbon containing molecules, that is a acetyl coenzyme A. And reaction oxidative decarboxylation of pyruvate to acetyl coenzyme A, because it is the carbon dioxide is releasing, it's called as decarboxylation. And oxidative decarboxylation, because here dehydrogenation also happening. So the protons are going to also will be released in the form of NADH. And coenzymes are TPP, lipoic acid, coenzyme A, FAD, and NAD. And this reaction also need to be tightly regulated. This is also this reaction pyruvate to acetyl coenzyme A also in a single direction. And this reaction will be uh, Irreversible reaction, but tightly controlled. It's controlling by allosteric inhibition or allosteric activation. Just now we have seen what is meant by allosteric, right? If the some other molecules bound to the enzyme to change its function, either activated or deactivated, that is called as allosteric modification. So allosteric inhibition is that this pyruvate hydrogenase will be inhibited by binding to acetyl coenzyme A. It's understandable, right? Because pyruvate is converted into acetyl coenzyme A by pyruvate dehydrogenase. When excess of acetyl coenzyme A are there, so they are going to bind to this enzyme and inhibit it. At the same time, NADH, if excess of NADH also going to be inhibit, or excess of ATP also inhibit. But in contrast, activation is by AMP. AMP molecules bind to the pyruvate dehydrogenase allosterically and change its structure to make more activated. So this is the outline of the this class that we have seen that the glycolytic pathway and state the energy required and energy producing steps in the glycolytic pathway, describe the regulation of the glycolysis pathway and explain the metabolic fate of the pyruvate. These are the things you have learned in the last class and today class together. So by this, you should be able to understand the glycolytic pathway, glycolysis whole process, and energy required for the priming step, and what are the energy produced in the other uh, other part of the glycolysis. And also, you know how the glycolysis process whole thing is regulated, mainly the three steps: the glucokinase or hexokinase, then phosphofructokinase, then pyruvate kinase. These are the three enzymes are regulating the whole glycolysis. And metabolic fate of the pyruvate is three ways. One is in aerobic condition, it will go acetyl coenzyme A. In anaerobic condition, lactate. In some of the organisms like microbes, it will go convert into anaerobic way. It will convert into ethanol, that is called as fermentation. And pyruvate will be converted into acetyl coenzyme A in the aerobic condition, and where it releases, it catalyzes the reaction by pyruvate dehydrogenase. This enzyme is a big complex enzyme, and this enzyme required 5 different coenzymes, and during this step NADH will be formed and COT will be synthesized. So this is the second part of the glycolysis. Let me resume the recording. Okay. This one, I told you, right? So that is the normal glycolysis process, 1,3-bis phosphoglycerate to 3-phosphoglycerate by the enzyme phosphoglycerate kinase, and the ATP is synthesized. But in RBC, what happens, sometimes this 1,3-bis phosphoglycerate, it convert into T3-bis phosphoglycerate by bisphosphoglycerate mutase, and then it will be converted back to 3-phosphoglycerate. This step is skipped, and the conversion of 1,3-bis phosphoglycerate to 3-phosphoglycerate will be handled in the 2 different steps by these enzymes, where ATP is not synthesized. The instead inorganic phosphate will be released. The bisphosphoglycerate mutase, this one, and 2,3-bis phosphoglycerate phosphatase, this one, is catalyzed by a single enzyme, bisphosphoglycerate mutase. So these two together a big enzyme. But at alkaline pH, this bisphosphoglycerate mutase favors the mutase activity, so that it will convert to 3-bisphosphoglycerate, while at lower pH, bisphosphoglycerate favors the phosphatase reaction, this reaction. So low pH and high pH is important for conversion to be present to be the molecules 2,3-bisphosphoglycerate in the cell or not. So what you understand with this step, the takeaway message is pH of the cell, especially RBC, will decide whether the 2,3-bis-phosphoglycerate to be there in the cell or 3-phosphoglycerate in the cells. So when it is alkaline, it will 1,3-bis-phosphoglycerate produce 2,3-bis-phosphoglycerate. In acidic, it will convert into 3-phospho. This you try to keep in your mind. We'll come to this step later. So let us try to understand some of your physiology, whether you may have heard, studied in a circulatory system or respiratory system, what happens, how the oxygen will be supplied from lungs to the all part of the body. So what is the major molecule required for it to supply the oxygen to the all parts of the body is hemoglobin. This is the hemoglobin. Is an average. Each red blood cell contain 270 millions of hemoglobins, and an average adult having 5 million red blood cell per microliter of the blood sample. So that many millions of hemoglobins are there. This hemoglobin, if you see the structure, the globin part having 4 proteins, 2 alpha chains and 2 beta chains, and they are arranged such a way which has shown in the picture. So hemoglobins adapt two different quaternary structure. This is called as quaternary structure. There are two quaternary structures, slight variation will be there. So one is called as T state, another one is called as R state. So what are these two different? When the hemoglobin is carrying the oxygen, when able to bind to the oxygen, it have a slight structural difference in the quaternary structure of the hemoglobin, that is called as R state. When the hemoglobin is not binding to the oxygen, then it is called as T state. Try to remember, that is the only thing you can remember. So what is that? It is having two states. When it is binding to the oxygen, it is R state. When it is not binding, T state. That is a slight variation in the hemoglobin structure. This switch between R and T state allows the release of oxygen in tissues and upload the oxygen in the lung. So that change is very important. See, when the hemoglobin is binding to the oxygen, and when it is hemoglobin coming to the lungs and binding to the oxygen, this is very much needed because oxygen need to be taken. And the same hemoglobin flow through the blood and reaches to the tissues where the same hemoglobin need to be released the oxygen, isn't it? So the affinity need to be changed. So if it is having the still same affinity, then it is not going to be useful. So at that time, the oxygen need to be released and gives to the tissues, and that means affinity should be reduced. And the same hemoglobin again goes to the lungs. It should increase the affinity. It should not have low affinity. If it is having low affinity, it cannot carry oxygen. So the switching of this R state to T to R is very important to change its affinity towards the oxygen to carry the oxygen to the tissues from the lungs. Okay, that is what here I would like to stress. Then hemoglobin will carry 4 oxygen molecules. Okay, 4 oxygen molecule. See, when first oxygen hemoglobin bind to the first oxygen forms oxyhemoglobin, the rate of reaction is quite low. Okay, and then once it get oxygenated, so binding to the second oxygen is quite fast compared to here, and to the third one is also very high, much faster, and fourth will be very high affinity when it is already 3 binding. So first one is having low affinity, second comparatively high, and third is much high, fourth is very high. So that's why you can see here that is called as cooperative binding. Once it bind, it will increase the affinity. So this is the curve shows between the oxygen um, availability, that mean how much is the oxygen present and what is a percentage of the sites filled. So when it is low oxygen, it will be very low, the binding to the hemoglobin, and it is trying to high slightly, and it will bind to the many. See this, you can see very logarithmic kind of change, and very high peak. Then once it reaches around 80, so almost 100% of the hemoglobin bind to the oxygen. Okay, so hemoglobin oxygen binding. When oxygen pressure is the different from 0 to 100, what is the binding sites filled in the hemoglobin? So for example, here you can see when it is around 40 is the oxygen pressure, that is what you can see in the tissues, right? In the tissues, oxygen utilized everything, so oxygen availability is very low. When oxygen availability very low, and during that time the binding sites filled with the hemoglobin is still 70%, even 40% of the oxygen available, 40 mmHg available, but 70% of the hemoglobin is binding. And when it is 100, what you can see in the lungs, so fully available the oxygen, the arteries, so where 100% filled. Though there is a huge change in the oxygen pressure, that means 100 to 40, and this the oxygen binding affinity will reduce slightly, that is 100% to 70%. So why we are talking all this, you will understand slowly. So if in case you have a vigorous exercise where you utilize a lot of oxygen in your tissues, and it will drop your oxygen to 20 or something, or 25, see the drop in your binding sites. So high oxygen will be given back to the tissues, so binding will be only around 25 something. So that means 70 to 25, that means it is around 45%. Even the small drop of the around 15% of the 15 mmHg drop will release a lot of oxygen from the hemoglobin to the tissues. So that is what I wanted to show you. A slight drop also gives, it will give you the oxygen affinity will be changed. Okay, so this is the black one, which is the normal one in the middle, that curve. So that means around 40 mmHg, around 70% of the hemoglobin will be bound with the oxygen. If it is having a less affinity, lower affinity, what happens? So same 40 mmHg, it will bind to 50%. That means around 20% of the oxygen which bound to the hemoglobin will be released if this is having low affinity. Or if it is having high affinity, let's say it is having high affinity, a slightly high affinity. So same 40 mmHg of oxygen, it will increase the affinity. It will bind more oxygen, even though if it is less pressure of the oxygen, less availability of oxygen, hemoglobin bind to the more to oxygen, more than 80%. So it will give very less oxygen to the tissues. So this is another thing you need to understand. The next step is what happens to the tissues during the respiration. Peripheral tissues. When carbon dioxide is forming during the respiration, so that is what we have seen, right? Like in a glycolysis, one carbon, not glycol, after the glycolysis, during the pyruvate to acetyl CoA, carbon dioxide released. Even the Krebs cycle or citric acid cycle, carbon dioxide will be released. So all this carbon dioxide mixed with the water, makes the lower pH because carbonic acid will be formed, or lactic acid also will be formed anaerobic condition. Whatever it is, it gives the acidic condition in the peripheral tissues. Okay, during after the respiration process, what is happening? It will produce lot of acids, and it is having a low pH. And what happens to the lungs? During in the lungs, so this carbonic acid releases as a carbon dioxide and water through the lungs. So thus it will increase the pH. So no protons there, much protons there. So the lungs area pH will be very high. I mean, not very high, high comparatively. So, so this, the graph which I have shown before, the black color one is a normal. That means oxygen when it is around 40. What is the affinity? When it is shift to the affinity, what happens left and right? So this is what happens when pH is slightly low. That is normally 7.4 is a physiological pH. When it is pH is 7.2, just slightly low, that means acidic pH. It's not acidic, comparatively acidic to 7.4 in peripheral tissues. What happens? It will change the curve to low affinity. That means the even the low oxygen, it will release more oxygen to the that particular place. Okay, so oxygen will be released in the acidic condition. So tissues will have more oxygen. So hemoglobin releasing the oxygen more. So when the same hemoglobin go and reach the lungs, lungs is having slightly high pH, that is 7.6. At higher pH, it's the affinity of the hemoglobin to the oxygen will change, and thus the same 40 can gives the less oxygen. That means tightly bound the oxygen during that time. So but anyhow, during in the lungs it's not 40, it will be much higher, almost like 100. So it will bind tightly to the oxygen, to the hemoglobin. So it will have 100% binding sites filled with the oxygen. So that is the pH will plays a significant role in the affinity of the binding of hemoglobin with oxygen. This is normally what we can see in the respiratory metabolism or physiology. So why we are talking all these things, how it is linked to the glycolysis. This 2,3-bisphosphoglycerate, which we have seen, especially in the red blood cells, conversion from 1,3-bisphosphoglycerate to 2,3-bisphosphoglycerate, it will lower the affinity. That means at low oxygen thing, so it will the hemoglobin lower the affinity to oxygen. That means it will releases the more oxygen. So binding affinity is low. So oxygen more will be released to the tissues, especially in the tissues, peripheral tissues. So when the red blood cells is going to the peripheral tissues, it will prefer to have the conversion from 1,3-bisphosphoglycerate to 2,3-bisphosphoglycerate inside the RBC, where the hemoglobins are there filled with oxygen. All will be released the oxygen during that time. So thus the tissues will be having more oxygen. When the affinity of the hemoglobin to oxygen is less, so oxygen more released, so it will be needed for the tissues. So that is how the glycolysis plays very important role in the respiration process. So then this one I already told, pyruvate dehydrogenase. What happens to it? It will convert into acetyl coenzyme A. So this pyruvate, which is converting into acetyl coenzyme A, catalyzing by pyruvate dehydrogenase complex. As I told you, it's a very big complex which is having three subunits E1, E2, E3. There's three enzymes together. It will form as a one big enzyme, pyruvate dehydrogenase complex. Pyruvate dehydrogenase decarboxylase or dehydrolipoyl transacetylase, dehydrolipoyl dehydrogenase. Separately, if you call, there are three different enzymes, but all together form a big complex that is called as pyruvate dehydrogenase complex. This complex requires five different coenzymes: TPP, lipoic acid, FAD, NAD, coenzyme A. Okay. So these are the, no, this is the complex E1, E2, E3 together, pyruvate dehydrogenase complex. This required coenzymes E1, thymine pyrophosphate, sorry, thymine phosphate, and then E2 subunit of the pyruvate dehydrogenase complex require two coenzymes, coenzyme A and lipoic acid, and E3 of part of the pyruvate dehydrogenase required two coenzymes, FAD and NAD. And this one we have seen the regulation. So acetyl CoA and NAD, ATP, all three inhibit the pyruvate dehydrogenase. AMP will activate the pyruvate dehydrogenase. Okay, so this pyruvate dehydrogenase, which is required all these five coenzymes, right? What is the source of these five coenzymes? All coming from the vitamins. So you have studied the vitamins also, right? So thymine pyrophosphate is coming from the thiamine, which is vitamin B1. So the deficiency of the thiamine is due to the dietary or alcoholics, and then it will lead to the formation, the beriberi, and which is wet type with cardiovascular presentations like congestive heart failure due to the low ATP. What happens when not sufficient vitamin B1, so which is needed for the TPP, which is the coenzyme for the PDH. So if you're not having enough vitamin B1, it will lead to the low ATP, and it will lead to the this kind of beriberi. And dry type is a peripheral neuropathy. So another vitamin B3, niacin, is needed for the nicotinamide adenine dinucleotide, is another coenzyme for the pyruvate dehydrogenase, NAD. So if you have a deficiency of this vitamin B3, so which is needed for the NAD, so it leads to the pellagra, so that is diarrhea, dermatitis, and dementia, and also can cause the serious central nervous system problems. And another one is B2. B2 is the source for the FAD, is another coenzyme for the pyruvate dehydrogenase. And if they have a deficiency of B2, then it leads to the angular stomatitis. And B5 is the source for the coenzyme A from pantothenic acid, vitamin B5. And regulation, we have seen that the high energy state or high coenzyme A, acetyl coenzyme A, or high NADH, this symbol indicates the inhibition. So it will inhibit the pyruvate dehydrogenase. So high reactants like glucose or pyruvates are very high in the cell, or low energy state of the cell, or NAD are also high, ADP high, then it will activate. So arrow shows the activation. So this is the another representation of the same enzyme regulation. So phosphorylation, dephosphorylation. So what happens, the kinase of PDK kinase actually inactivates the pyruvate dehydrogenase. So this whole enzyme will be bound to the phosphate group. So phosphorylated one is the inactivated state, dephosphorylated one active state. So calcium is the one will initiate the dephosphorylation. So that is phosphatase. So it will activate, but the pyruvate or acetate pyruvate and all will inhibit the kinase, so it will make into activate state, or acetyl coenzyme A or NAD or ATP will activate the kinase enzyme to be get phosphorylated, so in order to make it into inactive state. So this is the outcome of this today, this topic. So the whole thing, if you summarize, you can able to describe the glycolytic pathway, energy requiring and energy producing steps of the glycolytic pathway, describe the regulation of glycolysis, metabolic fate of the pyruvate. Apart from that, metabolic significance. Just now, one of the metabolic significance we have seen that what is the role in the respiration and carrying and releasing the oxygen to the tissues. So it is very important role of the glycolysis indirectly, because one of the substrate of the one of the intermediate of the glycolysis play a role in the respiration, or uh, the affinity of the hemoglobin to the oxygen, which is very highly needed to pass the sufficient oxygen to the tissues. Then pyruvate dehydrogenase complex also we have seen, and regulation of pyruvate dehydrogenase complex. So yeah, that's it about today's class. The both topics we try to complete.